Today our video is a microscopy of an onion root prepared slide. Just to review, there's another video that walks through, but just as a review, the ocular, labeled number 1 there, is our eyepiece with a power of 10. We have objective lenses that we'll be talking about as we rotate through those. We're also going to look at how the stage knobs will move the slide around in this video. And we're going to look at adjusting the amount of light through the iris diaphragm that's coming up through the condenser. We're going to begin, as always, with our scanning objective, or our 4x objective. Once we have it focused, we'll increase to the low power objective, or the 10x. And at this point, we no longer will use the coarse focus, only the fine focus to bring it into view. Next, we'll rotate to the next objective, the high power, or the 40x objective. And again, only using the fine focus, no longer the coarse focus adjustment. In this case, I'm also adjusting, as you notice, the amount of light that's coming through the condenser. Now we'll increase magnification to the oil immersion objective, or the 100x, which does require oil between the microscope slide and the objective. And now you see that I'm using the stage controls. So the stage knobs or stage controls are actually moving the stage around, which in turn is moving the microscope slide, so that you see different parts of the slide through the objective and through the oculars. I'm also using the fine focus to just find the best possible focus for the view, and using the iris diaphragm to change the amount of light coming through the condenser. So the multiple images in your lab this week are just taken at different spots along this onion root. So as we move the stage around, we see the majority of those cells are going to be in interphase. We just see a darker colored nucleus, it's stained darker. However, we do see that there are some cells in different phases of mitosis as we search around in this onion root tissue.